Gene therapy is a new and experimental medical technique that has great potential for finding a cure to many genetic diseases. Two main types of gene editing are somatic gene editing and germline gene editing. Both techniques are still experimental, but one gene editing procedure is more ethical than the other. Somatic gene editing is more ethical than germline gene editing. I'm a genetics major at Purdue University, and I have learned about various gene therapy techniques in my courses. Gene therapy is a great new technique that should be used to treat diseases, but not for personal use in embryonic development. Germline genome editing is one of two main gene therapy techniques. H.T. Greeley states in a 2019 article published in the Journal of Law and the Biosciences that germline genome editing is the editing of germ cells or gametes. Greeley also states that edits to the germline will affect the genome of the subject and all of their future generations. One of the main advantages that could be achieved with germline genome editing is the treatment and cure of genetic diseases. Geingel et al. Explained in their 2017 article titled The Ethics of Germline Genome Editing that germline genome editing could be used to identify and prevent genetic conditions in unborn embryos before they develop, or in the case of in vitro fertilization, even before they are implanted in the uterus. Although it has the potential to prevent genetic diseases from being passed through generations, the ethics of germline genome editing has many people worried. Germline genome therapy is unpopular with many medical professionals because of the effects it has on future generations. Geingel et al. State that germline genome editing will have negative effects on future generations because of the possible complications that could arise. Germline genome editing could mess with the process of evolution that has been adapting humans for hundreds of thousands of years. If germline genome editing becomes readily accessible to the general public, it is also a concern that it could be used for gene editing beyond just deleting genetic diseases. The idea of designer babies is concerning to many people. These babies would have their traits picked out for them. Isaac Rabinow states in his 2003 article, Gene Therapy Ethical Issues, that eugenically altering the population may not be the solution that many people believe it to be. Although many medical professionals agree that this technique could lead to dangerous consequences, even more of the general public finds germline genome editing to be unethical. According to an article by Vahen et al. In 2021 in the Journal of Community Genetics, students are concerned about the ramifications of germline genome editing. Their main concerns were the creation of designer babies, impediments on nature, and possible societal conflicts of the if the technology becomes readily available. Another type of gene editing that has been gaining popularity in the medical field is somatic gene editing. This type of gene editing is done in somatic cells or body cells. Lee and Niakan explain in their 2019 article titled Human Germline Genome Editing that CRISPR-Cas9 is one example of somatic genome editing and is a promising technique that can help find a treatment for many genetic disorders. Somatic genome editing is similar to germline genome editing in the fact that both techniques can help prevent diseases like cancer, cystic fibrosis, Huntington's disease, and other genetic disorders. It differs from germline genome editing because the genetic alterations are not in the germ cells and therefore cannot be passed down to further generations. Somatic genome editing is a more ethical technique than germline genome editing. Vahen et al. Explain that somatic gene editing is seen as more ethical than germline genome editing by the general public. Additionally, medical professionals are even more willing to see how this technique can be used to treat genetic diseases. Rabinow states that 96% of medical professionals support such intervention to cure life-threatening genetic diseases. Although germline genome editing can be used to treat genetic diseases, people are more worried that germline genome editing can be taken steps further and lead to being used for eugenic purposes. Somatic genome editing cannot be used for this purpose because the alterations done to somatic genes cannot be passed down to future generations. The U.S. Department of Health and Human Services, in their 2019 article titled Somatic Cell Genome Editing Program, explains that limiting the edits to these somatic cells reduces the risk of having unintended editing-related changes passed down to future generations. Two types of gene editing, somatic genome editing and germline genome editing, were discussed and evaluated. It was determined that somatic gene editing is more ethical than germline gene editing because it does not have a potential to lead to eugenic editing and is more supported by medical professionals and the general public. Gene therapy should not be used for personal use in embryonic development. However, gene therapy could be the future of personalized medicine if used responsibly.